So concluding our series on cell respiration, we are going to talk about the electron transport chain and oxidative phosphorylation. So we went through glycolysis, we were in the cytoplasm, we took glucose and we did a bunch of steps and we turned it into pyruvate. And we got a lot of NADH, where we took hydrogens off glucose and we took electrons off glucose and we made NAD plus into NADH, right? And we said that was going to be important, but we didn't really discuss why. Then we took pyruvate and we took it into the matrix of the mitochondrion and we made it into acetyl-CoA. And then we went through the citric acid cycle and we got some more NADH and we got another electron carrier called FADH2. And we also said that was important, but we didn't really talk about why. So in this video, we're going to find out why. First, I want to go over a bit of mitochondrial anatomy because you might hear terms like the matrix and not really know what is meant by that. So the mitochondrion, you might see it drawn something like this. These little foldy things are called cristae. We've got an outer membrane, we've got an inner membrane, and then the spaces in between. This one is called the intermembrane space because it's between the two membranes. And the other one is going to be called the matrix. So the very inside of the mitochondrion is called the matrix. And between the matrix and the inner membrane space, across the inner membrane, is going to be where electron transport chain and all that takes place. So all those lovely electron carriers that we just filled up with hydrogens and we filled up with electrons, they are going to give these electrons to a series of 4 complexes. And each of these complexes wants electrons more than the last one, or has a higher reduction potential than the last one. So NADH is going to turn back into NAD+, FADH2 is going to turn back into FAD, and so those electrons go through these complexes. And what about the hydrogens? What happens to them? Well, in order to make ATP, which is a really high energy molecule, we want to make energy, right? And one of the ways we can make energy is called potential energy. And potential energy can come about in a few different ways. So let's say that there is a cliff and I am a person on the cliff, right? I have potential energy because gravity is telling me, come down here, come to the bottom of the cliff. And if I fall down, I can translate that energy from potential energy into kinetic energy. So here in the same way, we are going to try to convert one type of chemical potential energy into another type of chemical potential energy because we can convert energies like that. So the way we're going to create chemical potential energy, or a chemiosmotic gradient, is we're going to put all the protons, we're going to pump them up into the intermembrane space. And all the electrons are going to stay with the complexes, or kind of stay near the matrix. And these guys, they're like best friends. They really want to stay together, and we just separated them. So the protons are going to want to get back to their electron friends, and the only way that they can do it is through an enzyme called ATP synthase. And ATP synthase is going to harness all that potential energy that's slowly going away as each proton crosses the membrane back to the matrix, and it is going to phosphorylate ADP and make ATP. Now this kind of seems complicated, so we're going to break it down with what each complex is doing. Now we're going to talk about what the electrons do. All of the complexes that are shown in this diagram with the protons pumping up, that's what they're doing with the protons. So that's all the same. We're going to focus on the electrons here. So complex 1 is going to work with NADH, and what it's ultimately going to do is it's going to reduce this molecule called coenzyme Q. The other name for coenzyme Q is ubiquinone, and so it's going to reduce ubiquinone into ubiquinol. So if we look down here, this is a little farther. Please ignore the stuff about like complex 3. But ubiquinone, it's called ubiquinone because it has these ketone groups. So we have ubiquinone, and then when we reduce it, when you reduce something, you're either taking away oxygens or adding hydrogens. When we're looking at these kind of groups like carbonyl or alcohol, the way that the sequence works is that carboxylic acid is the most oxidized you can get. And then you have an aldehyde or a ketone, and then the most reduced is an alcohol. I mean, you can reduce it further into just like a plain old alkyl group, but here we're going to be reducing these ketones to alcohol groups for ubiquinol. And so that's where we're putting these electrons that we are taking away from NADH. And we're using some intermediates. We're using an iron-sulfur subunit, and we're going to reduce it, and then we're going to not reduce it, oxidize it. We're also using this flavin mononucleotide, which is attached to a flavoprotein. It acts like a cofactor, and it is going to get reduced, and it's going to in turn reduce that iron-sulfur subunit, and that iron-sulfur subunit is going to reduce coenzyme Q into CoQH2, or ubiquinol. So every single one of these reactions, what's happening is one thing is getting oxidized and one thing is getting reduced. Complex 2 is also going to be reducing coenzyme Q, but it's going to do it with FAD and FADH2. So we have a little bit of a primer step because, if you go up here, it's showing you FADH2 getting oxidized to FAD. And if you look down here, you might be like, what the heck, why are we starting with FAD? Well, it's because we have to look back at the citric acid cycle. So this is actually a component of the citric acid cycle where we had succinate and we turned it into fumarate. And when that happened, it happened because we were oxidizing succinate. And in oxidizing succinate, we were able to reduce FAD into FADH2. And so this complex 2 actually is playing a role. It's that enzyme that we saw in the citric acid cycle, succinate-CoQ oxidative reductase. And so now we're going to take that FADH2 now that we have it, and we're going to use that same old iron-sulfur protein. We're going to reduce it and oxidize our FADH2 back into FAD, and then it can get reused back here in the beginning if we want to reuse it. And then the iron-sulfur protein is going to reduce coenzyme Q into ubiquinol. So complex 3 is going to take that reduced coenzyme Q and it's going to oxidize it again. And this may seem really counterproductive, but basically what we're doing is we're passing electrons along between different intermediates, and eventually we want to get them to oxygen, which is why we breathe in oxygen. So we have coenzyme Q, we're almost at the end, but we have this one more intermediate, and it's called cytochrome C. And cytochrome C is going to get reduced via its heme group. So I know it looks kind of weird because we start with a plus and we end with a plus, but that number goes down. So if we're less plus, we're more minus, or we have more electrons. So 2 cytochrome Cs are going to get reduced by 1 coenzyme Q. Because you see how ubiquinol has 2 reduced groups. Cytochrome C, each one of them is only going to get 1 electron. So that's why that is. And then we're going to take this cytochrome C, and cytochrome C oxidase, like its name says, we are going to oxidize it, and we are going to finally get to our electron acceptor. Come on, Sophie. Acceptor. And we are going to take oxygen and hydrogen, and we are going to make water. And these hydrogens, they're H+, right? When's the last time we heard H+? Well, we heard that H+ goes through ATP synthase. So that's where these protons are coming from, is they're coming from all the protons that we pumped up in the beginning or throughout these complexes. They're coming down through ATP synthase, and they're going to react with oxygen via cytochrome C oxidase, and they are going to make water, which is one of our products of cellular respiration. So this ATP synthase uses a bit of physical force or physical properties. So these hydrogens, these H+, these protons are going to go through a component called a stator, and they are going to go into a rotor, which, like the word suggests, it rotates around. And the force that it creates when it's rotating around is going to be translated into the energy that it takes to phosphorylate ADP into ATP. And then we have our ATP, and we get something like 32 to 34 ATP per glucose molecule from this whole process when we do it aerobically. And remember when we talked about fermentation and how organisms in an anaerobic environment have to go without this whole oxidative phosphorylation, and they just go by glucose, or what we call substrate level phosphorylation. You're only going to get like 2 ATP per glucose molecule. So this is really, really essential, this process of oxidative phosphorylation, this process of creating this chemiosmotic gradient. Now, a quick reminder, if you're in a lower level class, you do not need to know all this crazy complex stuff. I'm inclined to say that even if you're higher up and prepping for the MCAT, I've never come across anything that makes you memorize what is in complex 1 versus what is in complex 2. But this information is here if you need it, and I hope it helps you understand the way that we're passing these electrons along through the different components to ultimately get them to oxygen and get them into water. And that's how it works. So we've done this whole process now. We went through glycolysis, we went through pyruvate oxidation, we went through the citric acid cycle, and we're finally taking all these components that we took from all these earlier processes, the NADH, the FADH2, the succinate turning into fumarate in the citric acid cycle, and we are going to use them in oxidative phosphorylation and get our final big count of ATP. So I hope that helped, and now you know cellular respiration.